and welcome back to Med School Radio, where we play your favorites 24/7. So we're now talking about osteogenesis imperfecta, and we're going to talk about the genetic defects that produce OI. Sequencing of genomic DNA has shown that most patients with OI have mutations in genes for the pro-alpha-1 chain, col1A1, or for the gene for pro-alpha-2 1 chain, or col1A2, of type 1 procollagen, which is inherited in an autosomal dominant manner. Some mutations cause synthesis of a structurally abnormal but partially functional pro-alpha chain. These mutations include deletions within a gene and splice site mutations that impair the ability to precisely remove introns and link the remaining exons. Frequently, these mutations introduce premature termination codons, or PTCs, which trigger removal of the remaining abnormal mRNAs or synthesis of shortened proteins. Nevertheless, the most common mutation, of which there are several hundred, substitutes a single amino acid with a bulky side chain for glycine, thereby disturbing the ability of the procollagen chain to fold into the triple helix. Because collagen is composed of 2 pro-alpha-1 chains and 1 pro-alpha-2 chain, if either component is abnormal because of a mutation, the adverse effect on the structure of the triple helix can be quite significant. Such an effect is termed a dominant negative mutation because the product of the abnormal allele interferes with the function of the normal or wild gene product. Dominant negative mutations are most common in structural proteins like collagen, in which one abnormal subunit disrupts the function of the final molecule, which in the case of collagen is a trimer. Another unwanted effect of the incorporating of an abnormal pro-alpha chain into a pro-collagen molecule is that by preventing folding into the triple helix, the cell degrades the abnormal pro-collagen through what is termed the unfolded protein response. This complicated process is aimed at protecting the cell from the accumulation of unfolded proteins by degrading them in cytoplasmic proteasomes. Recently, autosomal recessive defects in cartilage-associated protein, or CRTAP, and prolyl-3 hydroxylase P3H1 encoded by leucine and proline enriched proteoglycan have been shown to cause some cases of OI types 2 and 3, which are severe forms of OI, along with cyclophilin beta, a member of the peptidyl-prolyl isomerase family. This complex of 3 proteins carry out the 3 hydroxylation of a limited number of praline residues in types 1, 2, 3, and 5 collagens. CRTAP is required for P3H1 activity, but is not itself an enzyme. It is quite remarkable that in type 1 collagen, the type found in bones, only 1 proline residue at position 986 undergoes hydroxylation by this complex.